Thank you for the invitation to present on the topic of evidence to support pediatric approval through extrapolation, focusing my reflections on the pediatric plans. Here's my disclosure. The views in this presentation are mine and are not the position of Johnson & Johnson, where I have the privilege of being a full-time employee. After setting the context, I will start with exposure matching, followed by the extrapolation of safety, matching response, the need for exposure response curves, extrapolation and controlled trials, and then some concluding remarks. The extrapolation concept synthesizes what we know and identifies the gaps in knowledge, including the degree of certainty or uncertainty about existing knowledge, which should be addressed in the extrapolation plan. The extrapolation plan must address the data required to establish both the efficacy and safety of the intervention, that is, the drug or biologic, for the indicated pediatric population. The categorical approach to the classification of extrapolation plans into either full, partial, or none is overly simplistic and will not be mentioned further, and should be replaced with a more nuanced understanding of how different plans may address the knowledge gaps which have been identified. If achieving a similar exposure is believed based on existing knowledge to produce a comparable clinical response, then finding the pediatric dose that matches the adult exposure should be sufficient to establish pediatric efficacy. This approach assumes that controlled pediatric data are not necessary to establish drug effect, whether based on previous pediatric trials or evidence that the pediatric control response would be similar to the adult response. In addition, this approach assumes that it is sufficient to establish an effective pediatric dose given the adult efficacy data, rather than an optimal pediatric dose through dose ranging, which may be ideal, but is not necessary to support pediatric labeling. Generally, exposure matching alone requires a pediatric sample size of between 12 to 15 participants for small molecules, and approximately 30 participants for large molecules based on the variability of the PK measurement. The required sample size may vary based on the inter- or intra-individual variability of the PK measurement and assumes sufficient homogeneity in the targeted age group with respect to key variables impacting on PK, such as absorption, distribution, metabolism, and excretion. This sample size will result in limited pediatric safety data, which may or may not be sufficient to address pediatric safety concerns, raising the question of the extrapolation of safety. Safety can be extrapolated from adults to adolescents and younger children with the same disease and exposure to the same drug or class of drugs, such as oral contraceptives or beta agonists for asthma. Safety can also be extrapolated from the same drug exposure in similar pediatric indications, such as psoriasis and psoriatic arthritis. Safety can be extrapolated across populations exposed to drugs with the same mechanism of action, that is, on-target effects. However, extrapolation of off-target effects may be more difficult. The requirement for specific pediatric safety data on growth and development should depend on the target population and the drug's mechanism of action, such as steroids versus beta agonists for asthma. Matching the pediatric response to the adult response may be useful if there's some uncertainty about whether matching the adult exposure alone would result in a comparable pediatric response. In this context, a pharmacodynamic biomarker may be useful in serving as a bridge in support of extrapolation between the reference adult population and the target pediatric population. For example, the N-terminal prohormone of brain natriuretic peptide was used as a bridging biomarker to extrapolate efficacy data from adult to pediatric patients with heart failure. The correlation of changes in the PD biomarker with clinical response should be established in the adult program, and the PD biomarker should be on the causal and/or mechanistic pathway of the drug-disease response. A PD biomarker may also be useful in reducing the required pediatric sample size if the variability in the PD biomarker measurement is less than the PK measurement, thus allowing for greater matching precision. Establishing an exposure response curve is not necessary for extrapolation. Exposure response curves only demonstrate a correlation between exposure and response based on their variability in factors such as body weight, and do not establish a causal link between a given exposure and the correlated response. Establishing a causal link between achieving a given exposure and a response would require randomization by exposure, not dose, a study of design which is not done for practical reasons. As such, the correlation of exposure and response is only indirect evidence of drug effect at best. Randomization to multiple doses may or may not increase the range of exposures available for such a correlation with response, but it does not change the nature of the inference that can be drawn from the data. Correlation does not establish causation. Exposing pediatric patients to a sufficiently low dose to achieve a lower range of exposures may be unethical if such a low dose fails to provide a sufficient prospect of clinical benefit to justify the risks, and thus raises the same ethical concerns about placebo use in some diseases. Extrapolation involves the leveraging of data from an external reference population, usually adults, but can be another pediatric population, to draw conclusions about the efficacy and safety of the drug in the target pediatric population. For pediatric trials involving a concurrent control, such as dose response, active treatment, placebo, or no treatment, external reference data can be used, in other words, borrowed, to reduce the required pediatric sample size. This reduction in the pediatric sample size will result in an elevated type 1 error, as with all forms of extrapolation, and should be justified by non-statistical considerations. For example, the clinical knowledge of the similarity of disease and treatment response, and the degree of tolerable uncertainty given disease severity and available treatments. Although there is not one standard for how much borrowing is justified, for example, exposure matching borrows 100% of the efficacy data from the target population. It is important to understand how much information is being incorporated to support the interpretation of the pediatric trial. For example, a tipping point analysis may be useful to understand how much weight needs to be placed on the reference data in order to draw a positive conclusion about the target pediatric population. Another approach is to use the effective sample size to describe how much information is being used. Ideally, the amount of borrowing should be pre-specified and sensitivity analyses performed to understand the operating characteristics, that is, the false positive or alpha and false negative or beta of the study design. Here's an example of a Bayesian tipping point analysis. On April 26, 2019, FDA approved belimumab for the treatment of children with systemic lupus erythematosus. The study was done in 93 pediatric SLE patients, randomizing patients either to Benlysta plus standard therapy or placebo plus standard therapy. The FDA found that based on discussion and feedback from the clinical team, it was reasonable to assume at least 55% weight on the relevance of the adult information to the pediatric population, and therefore concluded that there was at least a 97.5% posterior probability that belimumab was in fact effective. In contrast, taking a different approach but reaching the same conclusion, The European Commission extended to children 5 years and older the existing adult indication for belimumab as an add-on therapy for patients with active SLE. The EMA found that the clinical outcome of the pediatric study was comparable to the adult studies with a similar design, that the numerically higher response rate for the primary and most secondary endpoints was greater than placebo. And in subjects aged 5 to 11 years, there was no important difference in disease characteristics between children and adults, nor between children aged 5 to 11 and children older than 12 years of age. In conclusion, the EMA felt that the clinical data allowed for an extrapolation based on PK data. So this was using effectively an exposure response analysis approach. If failure to respond to the comparator or to drugs in the same class is an inclusion criteria, randomization to a comparator may be unethical and an impediment to enrollment. The use of an external placebo control group is a form of extrapolation in that it assumes the similarity of disease and treatment response, that is placebo, between the reference and target populations. When a concurrent pediatric placebo control cannot be used, for example, for ethical reasons, adding an active comparator offers limited, if any, value. The active comparator does not assure assay sensitivity, that is, that a comparable response by the investigational drug assures it is working, as that can only be provided by using a concurrent placebo control. Now for some concluding remarks. An extrapolation plan should be built on a strong foundation, that is, the extrapolation concept. Differences of opinion about the adequacy of the pediatric plan may reflect differences of opinion on the adequacy of the existing data, the knowledge gaps that need to be addressed, and the degree of uncertainty that needs to be dispelled. In other words, differences of opinion about the extrapolation concept. These differences should be identified and discussed directly by both the sponsor and the reviewing agency. Once there is agreement on the extrapolation concept, the design of the pediatric plan and analysis of the resulting data may be more straightforward, recognizing that there may be more than one approach to answering the identified questions. Thank you.